Good evening, everyone, and welcome back to our channel. Today we are discussing something interesting. We will look at the kingdom Monera and look at classification of the true bacteria and the archaea. Now, the archaea and the true bacteria are the two members in the kingdom Monera, and they are important. They look very similar in many ways, but they have a few differences which we'll just look at. It's a short video to just discuss archaea and eubacteria or true bacteria. What are their characteristics? What do they look like? And what are the important things to note about them? Now, the similarities between the archaea and the bacteria stems from their RNA sequences. The RNA sequences look quite alike and they look similar in structure, but they have slight differences, like I earlier said. Now, some of the differences include: one, the archaea do not have a cell wall. The bacterial cell usually have their cell wall. The archaea, they do not have a cell wall. Rather, they have what they refer to as a pseudopeptidoglycan layer. That's what you have in the archaea, which is not present in the bacteria. Now, archaeas are majorly what we call extremophiles, meaning that they survive in harsh conditions. Now, there are conditions that can be harsh for organisms. This includes conditions of extreme acid or base content, conditions of extreme heat, conditions of extreme salt content, even conditions of extreme pressure. We look at all these conditions and we refer to them as extreme conditions. And any organism that can survive and thrive in those conditions, we call them extremophiles. Extremophiles, so archaeas are extremophiles because they can survive under very harsh conditions. Now, archaeas, some of them are what we refer to as halophiles. And what are halophiles? Halophiles are organisms that can exist in conditions where there is very high salt content. Salt content is not good for many microorganisms, as it pulls away water and nutrients from inside their cells, and this makes them collapse. They become flaccid and die. So microorganisms, that's why we see that practical application of that is that a lot of preservation is done using salt, because it draws water content out of microbial cells and it destroys the microorganisms. So archaea can live in very high salt conditions. So some of them are called halophiles. We have those that are called thermophiles. Thermophiles are organisms that can live or survive and thrive in extreme high temperature conditions, like you have in volcanic eruptions, like you have in furnaces, volcanoes, all these things where you have very high heat concentration in that area. Organisms that can survive those kind of things are called thermophiles. Now, there are organisms that can survive extreme pH. We know what a pH meter is. We use that to measure the pH, either the acidity or the alkalinity of a medium. Now, there are organisms that can survive in very acidic conditions. We say those ones are basophilic or alkalophiles. And we have organisms that can survive in very acidic conditions. We call those organisms acidophiles. So archaeas are organisms that are unique in that they can survive in very harsh salt conditions. They can survive in very hot temperatures. They can survive extreme pH. It makes them unique. Now, a lot of archaea, an example of archaea that is very well known are organisms that are referred to as methanogens. Methanogens are very well known because they are organisms that produce methane gas as a byproduct of their metabolic activities. The methanogens are archaeas and they produce methane gas as a byproduct of their metabolic activities, making them very, very useful. Now, the methanogens can be found very easily in the stomach of ruminants, animals that are ruminants that have this four-chambered stomach. The goats, cattle, sheep, all these animals that regurgitate and chew the cud, they have methanogens, that's archaea, in their belly. So this is very important to know. Now, archaea, they exhibit what we call considerable genetic diversity, and they've been able to conquer a lot of environments, and they are present in a lot of environments. Now we can contrast them also with the bacteria, which are the true bacteria. The eubacteria are the true bacteria. They possess a peptidoglycan cell wall. We have a video on prokaryotes and eukaryotes where we discussed in details the cell structure of a bacterial cell. So that's what the structure of a eubacteria looks like, and we should go up and look it up. The peptidoglycan cell wall of the bacteria, that peptidoglycan is a polymer structure and is made up of peptidoglycan. Pep means protein or peptide, and glycan is for glucose. So that structure is made up of two major components, proteins and glucose. When we go further in this channel, we're going to look at the cell wall, the peptidoglycan cell wall of the bacteria, and discuss it in intimate details, what it does. Now, the bacteria can be found in many different environments. Bacteria can be found in soil, can be found in water, can be found on human body, can be found in different, it has also conquered different environments that are known to man, and they are surviving and thriving there. Now, bacteria also have a wide range of metabolic capabilities. That means it can do many things to survive metabolically. One of the things is that there are some bacteria that are photosynthetic in nature, meaning that they carry chloroplast and chlorophyll, and they're able to trap energy from the sun and use it to make glucose for their use. So an example of such bacteria are the cyanobacteria, and they are bacteria that are photosynthetic in nature. We have bacteria that are chemosynthetic in nature. These bacteria can make use of sulfur, magnesium, or any other chemical of their choice to use to generate or produce materials that they can use as their carbons, or as their source of energy, not carbon source now, as their source of energy. So we refer to them as chemotrophs or chemosynthetic organisms, bacteria. Now you also have bacteria that are heterotroph. That's these bacteria cannot, their mode of feeding or mode of nutrition is such that they need to obtain nutrients from other organisms. They can feed on other organisms. So bacteria that cause spoilage of materials, spoilage of food, what they are doing is feeding on the sugar content that is in these materials or this food which they are fermenting or spoiling. Now, bacteria have many things they do. Some bacteria are pathogenic in nature, meaning that they cause a wide range of diseases. Many bacteria are pathogenic in nature, and one must be careful with them. Some of them can even be transmitted from person to person. This is very important. But that's not the only thing that bacteria do. Some bacteria are mutualistic in nature, meaning that they are able to exist in mutually beneficial relationships with other organisms. And based on that, they are able to help those other organisms, and then they will also gain from the nutrients that are in those other organisms. There are many examples. You have bacteria that inhibit the gut of animals and even humans. For an example, bifidobacterium is a popular bacteria that is in the gut of human beings. Now, that gut provides a home, provides a safe haven for the bacteria, and is able to live there with causing minimal damage to the human being. And what does the human being gain from the presence of this bacteria? This bacteria helps to break down milk in the human being and helps to digest milk in the human being. So this is an example of a mutually beneficial relationship that can exist between a bacteria and a human being. Bacteria exist in many of such relationships with many organisms, and this is important to know. So we've come to the end of this class where we are just looking at archaea and bacteria. Look at the slight differences and similarities between two of them, as both of them share the kingdom Monera. Thank you for listening, and have a wonderful day.